Hello and welcome to this video on one of the oldest sources of bioactive compounds, plants. As a source of pharmaceutical products, plants have been known about for millennia. We have used them in a crude and unrefined format to treat injuries, disinfect wounds, and in religious rites. This is one of the naturalist arguments for the public right to all pharmaceuticals. That since many are naturally occurring in plants, there is no legal right to monopolize or control them. This point has been addressed before, but we will touch on it briefly in this video. As part of that history, there are older examples like opioids, and newer additions like aspirin and Taxol. These and other drugs have been discovered through a process of bioprospecting, which has shown us many novel and alternative drugs. We have also taken the plants and made the products better. To simplify the earliest history of medicine in humans, a caveman became sick while out in the field. They ate berries that made them ill, and they learned not to do so again. Meanwhile, the cave woman would add plants to their food to improve the color and taste. This would either make the food better or worse. As access to these possible foods increased, lessons were learned that eating some food in the right circumstances could make people ill or better. Some would aid in general well-being, while others were circumstantial. Some should be eaten in certain times of the year, and others never. Some should be eaten only by the elderly, the young, or specific conditions. This process went on until we reached a period of relative civilization. This involved agriculture and animal husbandry. The growth of populations accompanied this boom in agriculture and technology. More people meant more diseases that had never really been able to take hold before. This often led to death and mass infection. Attempts to control the spread of disease involved the use of religious rites to expel spirits and demons, a comforting procedure but of little practical value. Then we had the next big stage in medicine, that of the Hellenistic period. Admittedly, they were trying to treat the symptoms of disease, and it was not a huge leap forward. Wiping yourself with a goose will not solve hemorrhoids, nor will eating bread from the same loaf that led to you choking. They did get the notion of personal hygiene and regular bathing right. The Romans built on this with fresh water and regular food. But still, we did not have a strong program of medicine as it was developing in the West. There were still folk remedies and traditional medicines. These aimed to treat a causality that was called the four humours by countering them with the opposite product. After the Romans, Dark Ages, and medieval period, we have the Renaissance and the advent of Paracelsus, a man who coined the phrase, "the dose makes the poison." He was one of the first to scientifically approach the process of using chemicals and minerals as medicine, but even he had bizarre ideas. From here we did backslide with a period of quackery that involved bloodletting, histrionic diagnoses, and nephrology. We also developed many other treatments that are more recognizable as medicine. Quinine for malaria is a good example, and perhaps a decent place to start. Quinine is part of the bark of the cinchona plant, otherwise known as the quina tree. It was the mainstay in treatment for malaria from the 1600s through to the early 1900s. The tree had various names, and the one that should give away its long history is that of sacred bark. The various names were used from 1630 onwards, and were mostly given by Jesuit missionaries in South America. There are also legends of earlier uses by native populations. Both broadly follow the argument that a man became poisoned. Specifically, he developed a fever from malaria. He drank water that had been contaminated by the Jesuit tree. He then became better. Having shared his knowledge, use of the bark became common as a treatment, and extracting the quinine from it was used for a long time. No matter how it was discovered and who by, it found its way back to Europe, primarily starting in Spain. There, it was used as a treatment from 1638, over 100 years later, and in 1742, the botanist Carl Linnaeus called the tree the cinchona, after the Spanish version of that legend. Before 1820, the bark of the tree was first dried, ground to a powder, and then mixed into a liquid, generally wine, to cover up its strong taste. It was then consumed as a liquid. After 1820, the quinine was extracted from the bark, isolated, and used as a potent, ready-made medicine. This discovery was made by two men, Pierre-Joseph Pelletier and Joseph Caventou. The purified quinine became the standard treatment for malaria from there on. Extracting it from the bark of the cinchona tree was standard until 1920s. It was here that more effective semi- and fully synthetic antimalarials became commonplace. One of these is called chloroquine, and it more or less replaced anything that was occurring naturally from 1940s onwards. To reinforce this point, only the naturally occurring quinine was available as a treatment for malaria for hundreds of years. There was no other source, and it was very lucky that a cure existed in nature, that it was discovered, and could be used so easily. This is why we need a very diverse ecosystem. As with quinine, plants are the first obvious choice of finding new pharmaceuticals, as it has some of the easiest delivery methods straight into your mouth. At the same time, there are also a lot of plants, meaning a lot of possibly viable leads for new drugs. This is why mass projects exist that serve one purpose: extract possible products like quinine and test them against everything. They also describe the product's characteristics, structure, and possible ways to create it via synthesis. If it sounds like a lot of work, it is. This video is intended to go over just some of the steps involved in that process, and what is needed when we do not have the serendipitous luck that occurred with quinine. First, we need a plant. In this case here, we can extract something out of a piece of mint. In research, traditional medicinal plants are often chosen instead, but it could also be a crude, all things sampled over a given region. These are your prospective sources. Many projects will take that wide scope and focus on only one cluster, family, or specific sample of plants. This naturally limits the possible outcomes and will favor expanding the scope of research to more familiar or anticipated results. This could be the way one plant creates an antibiotic, and a similar species could produce a slightly different but more effective antibiotic. Some of the newer research highlights how we can better target and predict some of these characteristics based on the attributes of the plant and how they vary. Some grow in particular climates and soils, This can then play into the biosynthesis pathways and various other factors within the plant that will then affect the possible final products. It's a means of looking at one final intended result, such as an antibiotic for gram-negative or gram-positive bacteria that need different antibiotics, finding soil or environmental characteristics that have produced plants like that in the past, and looking in those areas for new plants that could have new antibiotics. A lot of the chemicals in plants are of little use, and this is one reason why you sample such a wide scope of them. The dominant chemicals found will be water and cellulose. Water can be removed from the equation by drying the plant matter. This is then used as is or turned into a powder. Cellulose needs to be chemically separated from your target chemicals. This is done several ways, but the simplest is a choice of solvents. As with past videos on the isoelectric point and high pressure liquid chromatography, each chemical in the complex environment of a plant is made up of varying charges. These can be pulled on differently with different solvents. Reverse osmosis water, methanol, ethanol, and other solvents like acetone will draw out and on two very simple differences in your plant matter. Water is very nonpolar and will draw out relatively nonpolar compounds, but it will also cause diffusion of everything. The more nonpolar or polar your solvent is, the more refined and specific your extraction can be. This is a process that can cover everything from the bark, leaves, buds, flowers, roots, and more. Think about the different parts and times that you can test a plant, and how those parts might change over a year, and then multiply that number by hundreds and thousands to account for just a small fraction of the possible crude extracts you may be getting out of this. Our mint example was extracted with a simple 20% ethanol solution, but it has pulled out many of the flavors that could be of interest. It is, by any stretch of the imagination, crude. Crude means that a little bit of everything is mixed into the extract. They exist in small quantities, and finding any kind of sense in this is not going to be easy. It can be compared to sifting out gold from a river of rubbish. You have also only extracted the compounds that correspond to the polarity of your solvent. This can leave everything that is not like your solvent behind. It may even leave anything that is not close enough to the polarity of your solvent, which is great for more refined extractions that target specific compounds. Depending on how much you have in your solvent, it may also be saturated. This means that you cannot extract any more of your bioactive compounds, even if there is more in the plant. This is similar to how tea can only get so strong in a cup or pot due to the size and quantity of water and tea leaves. Adding more solvent or more plant matter will only lead to the more soluble parts being extracted in greater quantities, but not necessarily a better overall extraction. This will generally extract the most common of active compounds in plants, starting with phenolics. These are very efficient interactors and antioxidants. Then you could find that there are glycoside compounds, which are another important and major group of bioactive chemicals. They are what make up hormones, sweeteners, alkaloids, flavonoids, antibiotics, and more. With your crude extract, you can begin performing your first and very preliminary tests. Microbes and cells for the most part. This helps to narrow down application or function. This is often in vitro, as any in vivo or animal-based studies tend to be expensive, time-consuming, and often run into trouble with ethics. Take this bottle of crude extracted mint essence. You can take this and get useful products as is. One of the simplest is to use chromatography to separate the contents into more specific groups of chemicals. It is a way to spread out the spectrum of your extract's polarity out further and further. Each time you take a sample of this extract, take out some and run it again, you can spread that sample out over an ever-widening spectrum of polarity, which means each chemical is more distinct and can be identified or pulled out of your crude extract individually. More often than not, it is column chromatography or thin layer chromatography. They are convenient, relatively cheap, and available in many different formats, whether that is silica, cellulose, and more, that give great value for money and are extremely efficient. This means you can train someone to run this sort of testing for very little and get very good results very quickly. Well before you even go so far as to consider trying to pull one of your chemicals out of all this mess, using all this equipment, reagents, and time, you need to see if the bioactivity you observed previously is still true, now in a more refined sample, or did you lose it into another fraction? Is it too dilute? Or was the effect a consequence of multiple chemicals interacting? After repeatedly refining, testing, and working on your extract, you will eventually get to a point where you have a relatively neat sample that has bioactivity that you desire. This is where the controversy over who owns it comes in. This refined, purified sample has not been invented, but the discovery of it and the means by which you get to that point are novel. This is one reason why it can and genuinely should be patented. The time and effort required to get to this stage far exceeds any reward that can be expected were there not a patent in effect. Eventually you get both enough of one compound and one that works in a model being used. A mystery molecule of sorts. A mystery molecule which needs to be identified in order to be patented and used effectively, but how? First, you have light spectroscopy. UV and infrared options exist. They are very good to begin with, as some molecules will be excited or respond to some wavelengths more than others. This allows you to narrow down the range. As an example, aromatic molecules are extremely powerful chromophores when hit with UV light. It is cheap, quick, and relatively easy, and in more refined and pure compounds, that is even more true. A small amount in a vial can be tested and retested, as it is a non-destructive process. This lets the longevity and stability of a possible lead be established. Then there is a more expensive and difficult process called nuclear magnetic resonance spectroscopy. This is primarily designed to figure out the magnetic properties of certain atomic nuclei. This means you can figure out where hydrogens are, how they are protonated, if there is a carbon in there, and some of the carbon isotopes. Once you start putting this into the bigger picture, and with a skilled chemist, you are able to understand how some of the various groups cluster together. These clusters then let you figure out what the positions of the nuclei are and how they go together in a very broad strokes picture. It will tell you how many atoms are present in each of these clusters, and past effort has worked towards using high-pressure liquid chromatography to isolate individual phenols in specific samples. The use of nuclear magnetic resonance spectroscopy allowed their structure to be elucidated, and in larger, more complex structures, you can use it to begin identifying active groups, small arms and clusters of molecules which could possibly be useful in a synthesis pathway. Then you have another option called mass spectroscopy. This is very useful for organic molecules. They are hit with electrons or lasers, and that allows each of the atomic elements within that to be converted to a charged ion. They then get thrown off at different rates and in different ways. The machinery used and the software then plots this as a relative abundance of the ion against the ratio charge of the ion. That tells you what it is. You can use this technology to determine with an incredibly high level of accuracy exactly what the entire molecule is made from. And depending on what's happened, when and where, you might even be able to figure out where the molecules came off from. Now you have one means by which a plant can be taken from the wild and possible leads for future drugs isolated, described, and have functions possibly identified and explained. More recently, we have commonly experienced bioactive compounds, primarily tannins. These are the bitter agents in wine, but also in part a dark coloration. These two properties could be useful as a staining product, but are not as useful in medicine. It is the protein precipitating property of tannins that have been useful. This is because it can function as an effective and strong antioxidant. The process of creating possible bioactive compounds is not limited to just pharmaceuticals. Extracting these sorts of bioactive compounds can be used in food science as a preservative, flavor, and stabilizers. They can be used as supplements to create fortified foods, even as a way to cheat in home brewing, as we have previously demonstrated. They are fundamentally everywhere, and we know so little about them and of them that the only way to find new drugs effectively is to scour the world through its biodiversity, identifying these new chemicals and possible uses. There are many other ways to extract the bioactive compounds, to refine them, and to test them, but this video was intended to give you a simple, single taste of a way it could be done. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.